Hello everyone, and welcome to phyloseminar.org, a project supported by the Society of Systematic Biologists. For this series of 3 talks, we are hearing from 3 individuals who have thought a lot about modeling heterogeneity in the substitution process across sites. Last time we heard from Andrew Roger, who motivated the need for more advanced models with evidence from deep phylogenetics and introduced the CAT models. Today we will hear from Nathan Whelan, who I believe will propose a site partitioning approach to handle the same issues. Finally, we'll hear from Nicolas Lartillot, who will describe Dirichlet process mixture models for site heterogeneity in detail. I want to remind people that the Q&A app is gone now, so you can now just ask questions through Twitter or IRC as described in the attending section of the PhyloSeminar website. If you have a different idea about how we can organize discussions, just email me. As I mentioned, today's speaker is Nathan Whelan. Nathan did his PhD at the University of Alabama in Tuscaloosa with Philip Harris. After that, he did a postdoc with Kenneth Helanich at Auburn University. He has just recently taken a position at the U.S. Fish and Wildlife Service, where he is director of the Southeast Conservation Genetics Lab in Warm Springs, Georgia. Welcome, Nathan, and thanks for participating.
All right, thanks, Eric, for inviting me to give a talk, and also thanks to the other people talking as part of this 3-talk series. So today what I'm going to be talking about is an alternative method for, I guess, modeling or accounting for substitutional heterogeneity in phylogenetics rather than just site heterogeneous models. Maybe perhaps you could see this as a, I guess, defense of most phylogenies that have been inferred, considering that the vast majority of phylogenies, even model-based phylogenies that have been inferred, have not been done with site heterogeneous models. And so I'm going to be looking at the relative performance of some different methods for kind of accounting for heterogeneity and how these models actually perform when it comes to inferring accurate branching patterns. So before I get started, I need to acknowledge a handful of people. Ken Helanich was my co-author on a paper we just had published in Systematic Biology, which is advanced access right now. A lot of what I'm going to be talking about today is in that paper, although there's one piece of new data which I think will be somewhat interesting. Kevin Kosat, Joey Cannon, and Milton Tan have kind of helped bounce ideas off of throughout the development of this project and this research. And then for that new piece of data, I should acknowledge some other co-authors, Lena Morose, Andrea Cohen, and Gustav Palle, and NASA did fund this research. So, I kind of approach this work from an empirical standpoint, and most of what I do is empirical phylogenetics. So a lot of what I'm going to be talking about is building on past theoretical work, and I should acknowledge the folks that have developed some of the more widely used programs and the programs that I'm going to be using, or that I use for a lot of this research. The Schematicus and his group, of course, developed RAXML, which I'm sure most people are very familiar with. It's one of the most widely used phylogenetics programs that's out there. Nicolas Lartillot, who's going to be talking for the next PhyloSeminar, developed PhyloBayes and the CAT models, and I'll be talking a lot about that today. So, thanks to him for all that theoretical development. And then Rob Lanphier and his group developed Partition Finder, which I imagine most people are also somewhat familiar with. And so I should acknowledge him because a lot of the work kind of builds off some of what he's done and really utilized Partition Finder pretty heavily in this research. And so ultimately what I'm interested in, I think a lot about what a lot of systematists and phylogenetists are interested in, is whether or not, one of the questions is whether or not empirical studies are actually obtaining accurate phylogenies. So we know that substitutional or site heterogeneity is present in real data, and we should account for that. And, there's certainly, a kind of a gold standard, maybe how you might model sequence data to perfectly capture the realities of sequence data. But of course, that would be a very complex model that might not be computationally intensive. So I was really interested in, are the methods that are currently being used in a lot of studies, are they actually obtaining accurate phylogenies? And so that's kind of where, how this project got started and why I kind of got interested in what I'm going to be talking about today. So Andrew Roger last week, or not last week, for the last PhyloSeminar talk, gave a really good description of a lot of the properties of sequence data. And so I recommend everybody, if they haven't seen that, to check out his talk. And I'll try not to overlap too much with what he talked about, other than say that it is widely recognized that substitutional heterogeneity is a property of empirical data sets. So this is just a simulated amino acid sequence. I don't think it represents any actual amino acid sequence, but it's just for illustrative purposes. And we also know that completely ignoring heterogeneity can result in inaccurate trees, essentially systematic error because the model is not describing the data well enough. And it's been described that that can happen, although it's not 100% clear, how often that occurs, but it certainly is something to take into consideration. And I certainly don't think that ignoring heterogeneity would necessarily be a good approach. So when I talk about ignoring heterogeneity, you can imagine a amino acid data matrix of many different genes. This is just a hypothetical example. You typically have, of course, larger genes and many more genes. And you might apply just a single site-homogeneous model to the entire amino acid data matrix. So that would be one set of relative exchangeabilities, one set of site frequencies, and one rate or gamma distribution for rate heterogeneity. And so that's applied to the entire data matrix. And so this would be an example of completely ignoring heterogeneity in phylogenetic inference. An alternative approach would be partitioning the data set and applying different models. So each model here would have its own relative exchangeabilities, its own frequencies, its own gamma distribution for modeling rate heterogeneity. And so you might partition up the data set in any number of ways. You could do it by genes, or you could possibly do it, combine genes in some manner. And there's methods now with Partition Finder, which I'll mention in a second, where you actually determine the best fit, partitioning scheme, and then the best fit models for those schemes. So in this case, gene 1 and gene 2 would have the same model, and then gene 3 would have a separate model. And then so, those models, of course, the kind of empirically derived site-homogeneous amino acid models, they have their own site frequencies that come with the model, but then you can also incorporate empirically derived site frequencies for those models as well. And so that would be, LG plus F or WAG plus F or VT plus F and so on and so forth. And so, kind of broadly about partitioning. So Partition Finder has become really popular for determining the best fit models and partitioning schemes. And I'll be using that program a lot in the kind of the analyses that I'll talk about today. And it's clearly become very popular. The 2 Partition Finder papers have been cited over 1,500 times. So it's clearly a method that's in wide use. Once you have those partitioning schemes and best fit model, you can infer trees with any number of programs under maximum likelihood or with Bayesian inference in a program like RaxML or Mr. Bayes or ExaBayes. Now, of course, there are some potential pitfalls to partitioning. This would be a very coarse method for handling substitutional heterogeneity. The first thing is that you have to define potential partitions a priori. So in Partition Finder, the term they use is blocks, feeding at blocks. So those blocks might be genes. They might be codon positions for nucleotides. Conceivably, if you have a well-annotated amino acid, you could even, or a protein, you could even add, partition it up, use blocks by protein domains. But then probably the biggest criticism for partitioning, and it's certainly a reasonable criticism if we're just trying to really model the substitution process perfectly, is that it is blind to heterogeneity within partitions. And I think that's certainly a potential pitfall. I think one of the things that we need to consider is, when is ignoring that kind of heterogeneity within partitions, does that really result in less accurate trees? And if so, when? So in contrast to partitioning, you have site-heterogeneous models. And so one way you might do this is give a single relative frequencies and a single gamma distribution for modeling rate heterogeneity to the entire data matrix, but then allow different categories to be assigned different site frequencies. And so this is just hypothetical. You can split this up in any number of ways. And so these sites in blue would have one set of site frequencies, and then these sites in red would have another set of site frequencies. And with an infinite mixture model like the CAT models, you could have any number of different site frequencies. And so the most widely used models site heterogeneous models are these CAT models, and they either assign the GTR matrix where the relative exchangeabilities would be derived from the data, or F81, you'll also hear CAT-F81, you'll also hear this referred to as either just the CAT model or cat-Poisson. And this is where you have equal relative exchangeabilities between amino acids for the entire data matrix. And so these models were developed by Lartiot and Philippe in 2004, and there's been a lot of work on these models. These are not the only site heterogeneous models out there. However, they are very widely used, and I was really interested in what empirical phylogeneticists are currently doing, and is that resulting in accurate phylogenies. Probably one of the reasons site heterogeneous models are widely used is that they are relatively computationally tractable, although even computationally tractable for a site heterogeneous model can kind of spiral out of control in terms of computational requirements. A lot of what I'm going to be talking about today, I think, is mostly in regards to these CAT models, but some of the things I talk about I do think are widely applicable to site heterogeneous models as a whole. So kind of getting to site heterogeneous models as a whole, they are usually implemented in a Bayesian framework, but there are maximum likelihood based site heterogeneous models. And of course, they're not blind to intergene heterogeneity, which, as we said with partitioning, that would be one of the downfalls, that it is blind to intergene or intrapartition substitutional or site heterogeneity. One of the pitfalls with these models is that they are very computationally demanding. It's been noted kind of anecdotally in a lot of studies that larger data sets simply will not run to completion. The Bayesian analyses simply won't converge in a reasonable time frame with the CAT models and other site heterogeneous models. But then another pitfall, which I would argue is a pitfall of site heterogeneous models and the CAT models in particular, is that the performance has not been well characterized in simulation. And while certainly a valid criticism of simulated data is that it's probably never going to be as complex as empirical data, I do think that understanding how models and techniques and methods perform in simulation is illustrative, and then we can use the results from simulation to look at empirical analyses in the context of that simulation. And ultimately, I think that provides a fuller picture of kind of the performance of different methods and models in recovering and whether or not they recover accurate branching patterns. And so I think it's important, and I've kind of hit on this, but to just focus in on this question before I kind of dig into some methods and results, and just think about why we might assume that trees inferred with site heterogeneous models would be more accurate than partitioning in the first place. And it really comes down to this fact that it's not blind to the site frequency differences within genes. But I think this comes with a major caveat. Just because a model is site heterogeneous doesn't by itself mean that as implemented, it's modeling that site heterogeneity well. And so I think that's something to consider, because if a site heterogeneous model isn't actually modeling the heterogeneity well, even if it can see within genes, that doesn't make it any better than partitioning necessarily. It's also worth pointing out that the site heterogeneous models and partitioning schemes with site-homogeneous models like WAG or LG or any number of models, they all assume base compositional homogeneity and don't account for heterotachy, which of course can also be model violations. And so that's at least something to just keep in mind when we're talking about these models. So despite this potential assumption that site heterogeneous models may result in more accurate branching patterns than partitioning, oftentimes analyses with CAT models or partitioning using site homogeneous models result in identical branching patterns when you actually infer an empirical phylogeny. And this is true for taxa ranging from Drosophila to land plants, so at different scales and on vast different taxonomic groups. However, there have been some pretty high-profile empirical disagreements. One of the empirical disagreements that I have the most experience with, because some of my empirical work while I was a postdoc, is relationships among non-bilaterian lineages, animal lineages. And so, early on when people were looking at this and some, from a phylogenomic with large data sets, typically CAT models would recover sponges as a sister lineage to all animals. And I think that this was taken broadly as evidence that the CAT models must be performing well because it matched preconceived notions about animal phylogeny, whereas other models, whether that be a single site-homogeneous amino acid model or partitioning, would result in ctenophores as the sister lineage to all other animals. And I guess, for me, it really came down to this question right here. Is this good enough evidence that cat models result in more accurate branching patterns? And perhaps, I'll come back to this later with empirical data as to why this may have been a questionable assumption. But I think more broadly, some of these more difficult phylogenetic questions probably shouldn't serve as, a major basis for arguing about model performance. And that kind of goes back to my perspective that we do need to analyze model performance in terms of recovering accurate branching patterns with simulation. Another thing in the literature, which I do want to kind of hit on and that I think is somewhat important, is that there have been studies that using Bayesian cross-validation in PhyloBayes to test model fit. And virtually always that shows CAT-GTR as being the best fitting model. And I'll get into why that might be. It's usually CAT-GTR followed by CAT-F81 and then, followed by an array of whatever site-homogeneous models may have been compared to. Generally, model testing with empirical studies with cat models simply is not done. One of the reasons that is probably the case is because it's quite computationally intensive to do this Bayesian cross-validation with these cat models in PhyloBayes. And maybe more to the point in terms of whether or not partitioning or site-heterogeneous models are fit the data better, is that currently there's not a program or a method available, or at least a program that implements a method where we can actually do model testing comparing the fit of cat models versus partitioning. So of course, when you see arguments that cat models are the best fitting models, that's based on Bayesian cross-validation, which I'm not saying is necessarily bad, but it doesn't compare cat models to partitioning. And considering that most phylogeneticists aren't using a single site-homogeneous model for a single amino acid matrix of 20,000, 100,000 plus amino acids, I think arguments about cat models fit the data better. It really hasn't been explicitly shown compared to partitioning. I do think there's an opportunity to develop some methods and implement that in some programs. It could conceivably be implemented in a program like RevBayes or modifying the PhyloBayes code, but I certainly think that's an area of potential development in the future. And I think it was at least just worth pointing out before jumping into the simulation studies. So I kind of said this a few times. I'll just harp on it again and jump right in to why simulation can be useful. And it really comes down to this simple fact that in simulation, trees are known. And with empirical studies, we can never know the true phylogeny with 100% certainty. Now, of course, a caveat of simulation studies is that it's difficult to explore all areas of tree space. We did try to look at some more complex trees that might be indicative or representative of some of the similar phylogenetic shapes that you see with empirical studies. So we did try to attack it from a more complex standpoint than the standard 4 taxon case. I think the 4 taxon case is illustrative, but it's not 100%. Well, it's not. I don't think that relevant in a lot of situations to the kind of data sets that most empirical biologists are encountering. But with simulation, it's still difficult to explore all areas of tree space. I don't think many people would disagree with that. And also, simulated data likely is never going to be as complex as empirical data, which is why I think it's important to do simulation studies, but then look at the simulation results in the context of empirical studies. And so, like I said, we simulated data on a handful of different tree shapes. These are a few. We used the R package PhyTools developed by Liam Ravel to just create a handful of pure birth trees that we could simulate data on. And then we also had 2 trees that had long branches and were susceptible to long branch attraction. The data sets that we simulated on them were susceptible to long branch attraction. We know that they were susceptible to long branch attraction because we ran maximum parsimony as a control. Of course, maximum parsimony is more susceptible to long branch attraction than model-based methods. And in that case, the long branches were attracted to each other. So we are pretty confident that these are trees that are susceptible to long branch attraction. And I guess the reason I'm most interested in looking at the simulated data sets that could be susceptible to long branch attraction is because site-heterogeneous models have been argued for very strongly because they purportedly suppress long branch attraction than other methods. So that was a feature of these techniques I wanted to look at. So we simulated each data set with 5 amino acids. I'm going to kind of briefly scan over this. It's all in the paper that we just had come out in Systematic Biology. We did try to simulate data. We simulated data with different size genes, number of genes, and different indel probabilities. And so those indel probabilities, by having indels that also kind of incorporated missing data into the analysis. Some data sets did have intragene heterogeneity. We did try doing posterior predictive simulation with CAT-GTR, and we determined it didn't really result in a data set with a reasonable amount of signal because we couldn't recreate with any model, including CAT-GTR, the relationships that the data were simulated under. So, I mean, I think that there might be some issues with just the way that's implemented right now in PhyloBayes, but I'll also talk about when I start looking at how CAT-GTR actually categorized the site heterogeneity, why posterior predictive simulation with the current implementation of CAT-GTR might be problematic. But we did at least give it the old college try with that one. So when we actually analyze this data, like I said, we use maximum parsimony as a control. We did do model testing with Partition Finder, so we didn't just feed it the perfect models and partitions. We let Partition Finder kind of do its thing and give us a best fit partitioning scheme and models. For partitioning, we used RaxML, and then with the CAT models, we used PhyloBayes and PI. The reason we didn't use the same program for both is because you can't implement partitioning in PhyloBayes and PI, and RaxML doesn't have these site heterogeneous CAT models. I should point out that don't confuse the site heterogeneous CAT models implemented in PhyloBayes with the CAT model in RaxML. The CAT model in RaxML is an approximation of for modeling rate heterogeneity, like the gamma distribution for modeling rate heterogeneity. So they're two different models with two different goals in mind. For PhyloBayes, we use default priors because you can't modify those with PhyloBayes and PI. And once again, that was what that's what people are doing with empirical data in empirical studies. We also took a look at, we ran F81 analysis with a single F81 model on data sets where CAT-F81 recovered incorrect relationships. And I think that'll become self-evident why we did that in a moment. But that was just to kind of explore whether or not the failure of CAT-F81 was a result of having equal relative exchange abilities across the entire data set, or whether the CAT component of that model was potentially causing some error. So to jump right into the results, and these are kind of broad, painting with a broad brushstroke here, but CAT-F81 often inferred low support for correct relationships. In all the trees that I'm going to show you, unless otherwise noted, the node had 100% posterior probability or bootstrap support. So a lot of the correct nodes have low support in this analysis, and then there was even an incorrectly inferred polytomy. We saw similar patterns with larger trees. And so this was a broad scale pattern for CAT-F81, where relationships were oftentimes incorrectly inferred, albeit with low support in this case, or the two cases I just showed you, and then correctly inferred nodes generally had limited support, at least less support than you might expect in simulation. In contrast, CAT-GTR and partitioning generally in simulation resulted in similar branching patterns and oftentimes very well supported branching patterns. And where they conflicted, much like what we see with a lot of empirical data sets, where they conflicted, the support was rather low. So in this case, we have a polytomy, which was essentially just a node with less than 50% posterior probability. And then with the tree inferred with RaxML and partitioning, also lowly supported with only 73% bootstrap support. And you can kind of see the nodes that are labeled there, where the nodes that are poorly supported with CAT-GTR are also less supported with partitioning as well. So similar behavior between these two techniques or models. Looking at these trees that were susceptible to long branch attraction, we did show with maximum parsimony that these data sets result in clear long branch attraction. On the other hand, with CAT-F81, we got an incorrect relationship, and it's inside this clade with high support where it shouldn't be. And it it's an incorrect relationship, but this taxon M was not a result. Doesn't look like long branch attraction. In fact, this long branch M taxon was pulled into a much more derived position. So keep that in mind, that CAT-F81 was pulled to a more derived position. That's going to be relevant when talking about some empirical data sets. And then CAT-GTR and partitioning, as we saw with the other data sets, resulted in accurate relationships. So at least with these data sets that were susceptible to long branch attraction, CAT-GTR and partitioning appear to be performing approximately similarly in inferring accurate branching patterns. But perhaps most troubling in terms of the performance of CAT-F81 was that when we use a single F81 model, and we saw this with some of the smaller data sets as well, the F81 model resulted in an accurate tree. This would seem to suggest that when CAT-F81 is inferring incorrect branching patterns, but partitioning in CAT-GTR is inferring relationships. But when a single F81 model is inferring correct relationships and CAT-F81 isn't, there seems to be something with the CAT aspect of CAT-F81 that is actually causing that error. And so what might be going on there is that these models are generally overestimating the amount of heterogeneity in the data set. And so we decided to plot out, how many amino acid characters were in a data set and how many inferred substitutional categories were inferred. So this is an average across the posterior distribution of the number of categories. And it generally overestimated, but maybe more troubling was that there was a highly significant, and in the case of CAT-GTR, rather good fit for just that the number of categories simply increased with the total amount of amino acid characters. Another thing that I think is worth pointing out here is that if you run a single data set with CAT-F81 and CAT-GTR, it's the same data set. The amount of heterogeneity in that data set has not changed. However, you'll get sometimes drastically different estimates of the amount of heterogeneity in the data set depending on whether you use CAT-F81 or CAT-GTR. So, I mean, ideally, a site-heterogeneous model, no matter what, relative exchangeabilities you couple with the site-heterogeneous component of the model, we would hope that it's still estimating heterogeneity in the same fashion. And it's a little unclear what really overestimating site heterogeneity will do to phylogenetic inference, but in the case of CAT-F81 in simulation, that could be the case, that overestimating the amount of heterogeneity in the data set actually contributed to heterogeneity. And I think this gets back to kind of that question of, even though these models are site heterogeneous, are they actually capturing heterogeneity well? It doesn't look like, at least with the data sets that we analyzed, that they are. And so in that case, they may be no better than partitioning in actually capturing the real amount of heterogeneity in a data set. I think it is worth pointing out that prior choice, particularly the hyperprior and the Dirichlet process prior, probably does affect the way categories are inferred, and site heterogeneity is inferred in PhyloBayes. We didn't look at prior choice because with the current implementation of CAT models in PhyloBayes MPI, you can't really explore prior choice, although I think this would certainly be an interesting thing to look at in the future. And then I would also argue, so one of the arguments against simulated data in the way that we did could be that the data set just didn't have as much heterogeneity as you might see in an empirical data set. And I think that's a valid criticism. And so one of the ways you might get around that is by simulating data with a site heterogeneous model, potentially with CAT-GTR and posterior predictive simulation. However, if it's wildly overestimating site heterogeneity, then those simulated data sets may be no more reliable or maybe even less reliable than the data sets that we simulated. And that might have contributed potentially as to why we were unable to get a reasonable signal when we did try to simulate data with CAT-GTR. Although I certainly think that's something that could be improved upon and looked at further. So kind of along those lines, we know that simulated data isn't ever going to be as complex as empirical data. So let's take a look at some empirical data sets. And as I said earlier, these models generally agree when you actually do phylogenetic inference with them. And when the results do disagree, incongruent nodes are often poorly supported. And maybe fortunately or unfortunately, some very high-profile data sets have disagreed in terms of the inferred relationships when using CAT models versus site-heterogeneous models with or without partitioning. So one example is the placement of Xenotrabella in Acelomorpha, and the data set I'm going to talk about the most is kind of non-bilateral animal relationships. And there's two reasons I'm going to focus in on this data set. One, it's the one I'm most familiar with because of my past empirical work, but also I would argue this Philippe et al. 2009 data set in particular has been used as a workhorse for postulating about the accuracy of CAT models. Because when they used a CAT model, CAT-F81, they recovered sponges as a sister lineage to all their animals. And this did appear, or this does kind of match kind of preconceived notions of animal phylogeny. But, based on some more recent analyses, I think that assumption is kind of called into question of whether or not this is the true phylogeny, this diagram I have here. So we reanalyzed both data sets with CAT-F81, CAT-GTR, and partitioning. Philippe et al. 2009 was only analyzed with CAT-F81 and a single site homogeneous model, not CAT-GTR or partitioning. So I wanted to see how those models performed on that data set. Nosenko et al., they did use CAT-F81 and CAT-GTR, but they didn't use partitioning. So I thought these were going to be good data sets to kind of look at how these models perform with empirical data. So very surprisingly, at least to us, was that CAT-F81, CAT-GTR, and partitioning all recovered ctenophores as a sister lineage to all other animals on this Philippe et al. 2009 data set. Now, this conflicts with their reported phylogeny. I'm not really sure what's going on there, but it could have to do with algorithmic or programming improvements in newer versions of PhyloBayes since the publication of Philippe et al. And you can see here, assuming this is the correct tree, CAT-GTR and partitioning are well supported. And then similarly to what we saw in simulation, CAT-F81 has lower support for a correctly inferred node. And I'll get to why I think this is probably the correct placement of ctenophores in a moment. With the Nosenko et al. Data set, we found a similar pattern with CAT-GTR and partitioning, where they both supported with nearly maximal support ctenophores as a sister lineage to all other animals. In contrast, CAT-F81 recovered ctenophores as a sister group to cnidarians and sponges as a sister lineage to all other animals. And while this does kind of match preconceived notions about animal phylogeny and kind of the historical perspective, I think if we think about how CAT-F81 behaved in simulation, it's rather illustrative. In that case, when it performed incorrectly on one method, or with one data set, it pulled a lineage to a more derived position. And that seems to be what's happening here in terms of the placement of ctenophores. So I think given that, we can probably say with some degree of certainty that the CAT-F81 tree is the less accurate tree. And in that case, it recovered ctenophores essentially in the wrong spot. I do want to mention something about model testing here. Nosenko et al. 2013 did do model testing with that cross-validation approach. And they showed that CAT-GTR fit the data best. CAT-F81 was second, and then some site-homogeneous models were behind that in terms of apparent model fit. However, the single site-homogeneous models actually got the placement of ctenophore sister all other animals more similarly to what we see with larger data sets, which much more taxon sampling. And so this is a new data set that we're going to be submitting very soon, a lot more ctenophore and some more sponge sampling than previous studies. And both CAT-GTR and partitioning recover ctenophores as a sister group to all their animals. And so that seems to suggest showing that some of these models may have fit the data less than CAT-F81. Maybe that model testing was a little problematic. I'm not saying that model testing as a whole shouldn't be done or that there's anything wrong with model testing at its core, but cross-validation as an approach hasn't been widely used for substitution models. And I think it deserves a lot more critical analysis, especially in terms of whether or not maybe it's potentially biased towards very complex models and not necessarily giving a full picture of what model is going to result in the most accurate relationships. And so I do think there's some work to be done there, and also some work to be done where we can actually better do model testing, comparing site-heterogeneous models to different partitioning schemes. Now, I haven't really shown any instance of where CAT-GTR really performs differently than partitioning, and so I want to kind of point out some examples where we see that with empirical data. Most of the other example I've shown you where there was disagreement was in regards to CAT-F81 versus other models. So in the Nimiopsis ctenophore genome paper, it was a really nice paper by Ryan and others that came out in 2013. In one of their analyses with CAT-GTR, they recovered sponges and ctenophores sister to each other, and then that clade as a sister group to all their animals. This is a relationship that hasn't been recovered in subsequent analyses with much greater taxon sampling. And so in this case, it would seem to suggest that CAT-GTR may have been contributing to some systematic error. And notably, a single site-homogeneous model recovered ctenophore sister to all their animals, whereas, which is the pattern that we're seeing with partitioning and now CAT-GTR on data sets with greater taxon sampling. And so I think that's an instance of possibly where CAT-GTR actually wasn't performing as well as some other techniques. There's also been some disagreement with CAT-GTR in regard to the placement of xenocoelomorpha. So two recent papers, two really nice papers, one by Joey Cannon and others that came out in 2016, and another one by Simakov and others in 2015, with partitioning and CAT-GTR recovered xenocoelomorpha as the sister lineage to all other bilaterians. In another paper that came out at the same time as the Cannon et al. Paper, this was Rouse et al. This was a paper some of you may remember from either reading it or for some of the publicity that it got, described some deep sea xenoturbellids, these purple sock-like deep sea worms. And so it was a really nice paper. And in one of their analyses with CAT-GTR, they recovered xenocoelomorpha sister protostomes. And so this is a relationship that hadn't really been seen before in maybe more complete data sets in terms of taxon sampling, especially the Cannon et al. Data set. They don't get that relationship with either partitioning or CAT-GTR. So that seems to suggest that the CAT-GTR may actually be influencing some systematic error occurring. And then the final example I'll show you is a paper I was involved with, Lee and others, including myself and Kinelonich, that just came out in Zoologica Scripta. And this was looking at Ciboglinidae relationships. And so this is a family of annelids. And this one taxon Spirobrachia, partitioning and CAT-GTR generally disagreed on its placement, with partitioning placing it in a clade with some other a clade of frenulates, and then with CAT-GTR placing it sister to all their Ciboglinidae. The thing is that the tree recovered partitioning Spirobrachia as a frenulate is a relationship that's supported by a fair amount of morphological synapomorphies and also some past mitogenomic studies. So in all these instances, it's impossible to know the true tree with certainty. But I think it's telling that generally when taxon sampling is high, partitioning and CAT-GTR do get the same answer. But in previous data sets where maybe taxon sampling wasn't as high and they disagreed, the tree that matches what's inferred with, I would argue, better data sets, since there's better taxon sampling, matches partitioning rather than CAT-GTR. And this really does go against the grain of what you see in the literature. The literature would seem to suggest that CAT-GTR is, in fact, incredibly good at suppressing systematic error, and in particular, long branch attraction. But there's certainly some instances where CAT-GTR and partitioning have disagreed, and it appears that the tree that was susceptible to error was actually the tree inferred with CAT-GTR. I should point out here, and I think this is worth more study broadly about site-heterogeneous models, is that Spirobrachia did have a fair amount of missing data. It does appear that when a single taxon or a group of close related taxon do have a high proportion of missing data compared to the rest of the taxa, that seems to be where CAT-GTR and partitioning disagree the most, and where the CAT models seem to do worse in terms of inferring relationships. Now, how missing data affects phylogenetic inference has been pretty widely explored, but not so much with site-heterogeneous models. And I think that is a feature of real data that we should take into consideration when we're testing not only the CAT models, but site-heterogeneous models and partitioning as a whole. So in the last talk, Andrew Roger had this quote that I pulled right from his talk, because I think it's actually a very great goal. in that we develop models that take in the true process of substitution, so we can get accurate phylogenetic reconstruction without having those models too complex. And so this really is a matter of trying to determining, is, for example, modeling heterogeneity in a coarse matter like partitioning, is that going to be good enough, or do we need site-heterogeneous models? And I think it's also worth pointing out is that just because it is site-heterogeneous models doesn't necessarily mean that its implementation is going to be perfect. And so it does need thorough testing, not just the models, but the software that implements those models when it comes to determining whether or not we're getting accurate phylogenetic hypotheses. So kind of just moving forward and kind of wrapping up my talk, I'm not biased, hopefully you don't think I'm completely against site-heterogeneous models. I think complex models and models that better model the substitutional process is very important, but models and methods need to be tested, and they need to be compared to what empirical phylogeneticists are actually doing. Comparing site-heterogeneous models to a single site-homogeneous model, while illustrative, if that's where we stop when comparing model performance, I would argue that's simply not good enough, because most empirical studies aren't just using a single site-homogeneous model for the entirety of a very large amino acid data matrix. Along the same lines, if we compare site-heterogeneous models to partitioning or any other method, we need to make sure that those methods are being widely used. For example, assigning a single relative exchangeabilities and a single site frequencies and so on, but then just partitioning branch lengths, that's really isn't what's being done in most studies. And so, if we're going to compare CAT models or site-heterogeneous models as a whole to partitioning or any other technique that somebody comes up with, we need to make sure that we're comparing them to what people are doing. And in the case of partitioning, this would be each partition having its own relative exchangeabilities, its own site frequencies, its own, so on and so forth. And kind of some broad recommendations. I do think that partitioning appears to be rather accurate. Are there situations where partitioning may fail? Absolutely. But clearly there's also instances where site-heterogeneous models will fail. And so, I think some of the tone of some papers and literature, really seem to come off that site-heterogeneous models are nearly infallible. And that certainly doesn't seem to be the case. And, I think it's also worth taking into consideration what is actually computationally feasible with, the computers that we have available. Just in terms of wrapping up results, CAT-GTR and partitioning did perform similarly in simulation. I think that's important. And I do really think that we need to look at how site-heterogeneous models are actually estimating site heterogeneity and whether they are overestimating or underestimating the amount of heterogeneity in a data set. Because if they're doing that on a problematic level, then that doesn't make a site-heterogeneous model any better than partitioning. So I kind of said that first one. I think that's important. No model is infallible. It's just a model. No model is perfect. Also, the models that I talked about here all kind of take into account or don't take into account base compositional heterogeneity or heterotachy. These may be bigger problems for some data sets than we currently appreciate. There's a really nice paper by Kosada and others that's also currently in advanced access at Systematic Biology. And they suggest that maybe base compositional heterogeneity is one of the biggest problems in terms of trying to accurately or confidently determine relationships among Lophotrogozoa. So, I do think that models that take these two things into consideration, assuming that they're computationally tractable, would really advance the field. And then just kind of suggestions. Hopefully these are things that everybody can disagree or agree on. I know some of the things that I probably said today in terms of the performance of site-heterogeneous models might be a little controversial, but I think that some of these things are a little less controversial. I don't think that CAT-F81 should be used anymore. The equal relative exchangeabilities was never really considered to be realistic, but it was an attempt to make the CAT models more computationally efficient. However, it doesn't really seem to be resulting in accurate trees, and we've shown that in simulation, and hopefully it was somewhat convincing with the analysis of those non-bilateral animal data sets. one of the things that people have started doing to, I guess, help with the computational demands of the CAT models is removing constant sites, or worse, parsimony uninformative sites. Parsimony uninformative sites are informative with model-based phylogenetic inference. And so just removing them because we want to get a model or an analysis to finish faster, I really don't think that's a good enough reason to be removing data, especially considering that I think the evidence that like at least CAT-GTR, which is very widely used, I think the evidence that it's that much better than partitioning is lacking. And so just removing data sets to get it, or data to get an analysis to finish without any other justification, I think is problematic. And then without calling out names or anything, a troubling trend that I've been seeing, especially when running site heterogeneous models, particularly the CAT models, is that people have been reporting Bayesian analyses that show no evidence of convergence. What will happen is convergence statistics are reported, and those convergence statistics don't show any evidence of convergence, and yet the trees are reported. I think this is problematic because those trees aren't statistically valid, and it's essentially stopping an analysis before it's finished. So hopefully we can all agree on that, and maybe more broadly, we should dig deeper into the convergence statistics with these site heterogeneous models, because we need to make sure that our analyses are being at least done in a robust fashion for whatever method we choose. And I should say too that I'm not against Bayesian approaches. I actually like Bayesian approaches. I think they've been a huge advancement in the field, but we do need to consider prior choice and convergence whenever we're doing Bayesian approaches. And of course, saying that, I realize I only use default priors for the CAT models, but it's a little difficult if the program being most widely used doesn't easily allow for modifying prior choice. So I think that's worth pointing out. And so hopefully we've reached some agreement on that. And so with that, I think that's hopefully I've convinced one or two people kind of my perspective on the situation. And I would just thank everybody for listening. And I think I have some time for questions.
Thank you very much. That was a very thought-provoking and clear talk. So I want to remind people that hopefully you have questions. There's two ways that you can ask questions. One is by tweeting at PhyloSeminar, and then the other one is by going to the IRC channel, which is #PhyloSeminar. And if you're not familiar with that, there's an easy way to get to that from the PhyloSeminar website. And so far, I've only seen Andrew Roger there. I think he had to leave, though, so he may not be able to ask questions now that we're at the end of the talk. Maybe I'll just start off the questions a little bit by asking a little bit more in detail about how the intragene heterogeneity was simulated in your talk. I mean, you've several times during the talk, including at the very end, you described how it seems like base composition heterogeneity and heterotachy and things like that are potentially important features of real molecular sequence evolution. And maybe for people who that haven't read your paper, you can describe a little bit more in detail about how you simulated those.
Yeah, well, so in our data sets, because we knew that both models that we were going to be looking at didn't, assumed base compositional homogeneity and a lack of heterotachy, we didn't actually incorporate that into our simulated data sets. In terms of simulating the kind of intragene substitutional heterogeneity, is essentially when we simulated any given gene or sequence with WAG or LG, what we did was we then ran analyses having those separate as different blocks when we fed it to Partition Finder. But then what we did was we took those same exact data sets and we essentially randomly combined the genes. And so a gene might be, have components of the WAG model and the LG model and the VT model all in that one gene. And then that combined, I guess, a sequence was used as a starting block for partitioning. And so that was the instance where we tried to incorporate, that intragene heterogeneity into, I guess really either you can think of it as genes or kind of blocks that we fed to Partition Finder. And so in that case, partitioning would have been blind to that intragene substitutional heterogeneity. And certainly, I guess
I'm just asking, I mean, like how many, like if you, a couple of PhyloSeminar sessions ago, we had Richard Goldstein, and he was talking about, pretty extreme levels of what he considers to be realistic intragene heterogeneity. So I mean, are we talking about a few genes getting mixed up or a lot of genes?
And we're talking about at max 3 genes getting mixed up, but then if the data set has 200 genes, there's 67 genes with intergene heterogeneity. So I mean, that is not as much heterogeneity as you might see in some empirical data sets. I completely acknowledge that. I think that's just going to be a feature of most simulated data, which is why I think it's important to also look at some of those empirical data sets.
Yeah, I mean, I think you've done a great service by doing simulations. I mean, it seems like it wouldn't be that much harder than what you did to just try simulating under more extreme levels of intragene heterogeneity, which has been suggested by different people. But anyway.
So I think that's 100% correct, and I certainly think that it would be valuable to do more analyses in simulation with more complex sequence data sets. And we were kind of limited on what we could do just because our study, we used over 76 CPU years worth of computational time. 97% of that was done exploring these CAT models. So we were kind of limited on what we could do in terms of having something that we thought was worth getting out there and publishing versus spending literally years trying to analyze more and more complex data for a single publication. But I certainly think that would be valuable to do in the future.
Cool. Yeah, I'm sure that there'll be continuing to be more discussion about this. So Andrew Roger says, interesting talk. Your simulations were based on different empirical substitution matrices applied to different partitions in the data, yet we, Wang et al. 2008 and Lartio, have shown that such models will generate data that is more uniform amino acid frequencies at sites than real data. But this setting is a case in which CAT-F81 is violated, but your partition models are correctly specified. So to some extent, it is not surprising that the latter perform better. So it seems like a fair simulation study would generate sites with less uniformity in frequencies like real data. Thoughts?
I mean, I think that's a valid criticism that the simulated data could be more complex. Two things along those lines. The first one would be that I think that our conclusions hold up in the light of a fair amount of empirical analyses with empirical data sets. But then more to the point, I think maybe one of the reasons it was worthwhile to look at some slightly simpler data sets was to kind of get at how these different methods are actually accounting for that site heterogeneity. And even with a very limited amount of site heterogeneity, the CAT models seem to be really overestimating it. And I think the way that it just scaled with data set size rather than the true amount of heterogeneity, I think that's pretty problematic and something that just hasn't been explored enough in depth. And so for that reason, I think I think it was valuable to look at simulated data. But like I said, I think that there's work to be done on more complex data. But if we do simulate that data, I would emphasize that we should try to simulate data if we're trying to get more realistic. That we're also not wildly overestimating the amount of site heterogeneity in data sets. And it certainly seems if we use the CAT models, at least as currently implemented, to simulate data, we would be kind of overestimating site heterogeneity as well. So that was another kind of consideration that we kind of thought about.
Yeah. Hey, Nathan, why don't we just turn on, I mean, I don't see any questions that are going to be needing your slides, so why don't we just go ahead and turn off the screen sharing so we can have a
That's
bit
fine.
more
Yeah.
Conversation.
I got to do that on my end, don't I? Let's see. So I just stopped slide sharing?
Yeah, stop screen sharing on the Google Hangout window. So while you're doing that, I wanted to ask, hello. So over Twitter, Kevin Coscott asks, do you have a strong feelings for using partition finder rather than e.g. the RAXML auto model selection?
I guess my perspective on that is that I think RAXML, based on my experience, does a pretty good job with model testing for single gene alignments. The problem is, at least as far as I'm aware, it doesn't allow you to combine genes that might have similar substitutional profiles like you can with partition finder. And so in that instance, for that reason, I'd probably prefer the model testing with partition finder for larger multi-gene data sets.
Cool. P. Simeon, I'm not sure what his actual name is, would like to ask a question I'd like to ask for Nation Whelan. The accuracy of site heterogeneity modeling of CAT has been demonstrated in terms of categories of amino acid profile in its first publication. How the use of partitioning can improve that aspect when using site-homogeneous models?
So, if I understand the question correctly, I think kind of the answer to that, and maybe I didn't explain this as well. I'm not saying that partitioning perfectly models the way proteins evolve. It probably doesn't, but I think what I was I was really getting at is whether or not that method, since it is computationally tractable, is it resulting in accurate trees? And then comparing that to some of the more popular site heterogeneous models. And so, I've certainly acknowledged that partitioning is not going to be a perfect description of amino acid evolution, but in terms of obtaining accurate trees, it seems to be doing a pretty darn good job.
Cool. Yeah. So Andrew, Andrew is just kind of continuing about asking about further means of simulation. So he suggests, how about simulating under empirical mixtures like Lee, GASQL, and LARTO, estimated by maximum likelihood on a database, the ones with 10 to 60 site classes, or the secondary structure classes that GASQL has published?
Yeah, I mean, I think that would be a good good way to take a look at modeling data or simulating data in a more realistic fashion. I mean, I think that as a field, we'd actually benefit from better techniques and accessible techniques for modeling more complex simulated data. But I do still think there is value from taking a look at relatively simple data as well in terms of simulated data.
Yeah, I think more advanced simulations is something all fields really need. I mean, I really was struck by it wasn't just on the simulated data, but the ways in which, at least in your hands, the CAT-F81 and the CAT-GTR models had really different behavior. I mean, I guess it is those exchangeabilities like you're talking about.
I think that's got to be playing a pretty large role in it. I mean, I guess for a 20,000 plus amino acid sequence data set, I'm not sure that we would be expected to maybe we shouldn't have expected that in all situations adding site heterogeneity is going to save equal relative exchangeabilities from itself.
Yeah. Okay, well, I'm not seeing any other questions. Thank you very much for the fascinating talk. The next talk is going to be on November 16th, and that's going to be from Nicolas Lartio. And I look forward to hearing that. And thanks again, Nathan.
Yep, thank you.